Hey everyone, welcome to our research project presentation for the ThinkNeuro Spring 2024 Clinical Research Internship. In our presentation, we will be discussing therapeutic interventions within the pathogenesis of tauopathies, and we're very excited to share our findings using bibliometric analysis with you all. First off, we'll be introducing ourselves. My name is Prithika Poomaran, and I am the Research Associate for the Tauopathies research team. I am currently a first year at UC Berkeley studying microbial biology, and I'm on the pre-med track.
Hi everyone, I am Kayla Chen and I am the Research Co-Associate. I am currently a third year undergraduate studying economics and neuroscience at UC Riverside.
Hi, my name is Vidhi. I'm a sophomore at California High School in San Ramon, and I'm intending to major in neuroscience or cognitive science. I decided to join ThinkNeuro because I'm extremely interested in neuroscience and I love learning new things about the brain and the human body, and I thought this would be a great way to learn some new things.
I'm Jackeline and I'm a junior from Scripps High School in San Diego, California. I'm thinking of majoring in neuropsychology or microbiology, and I wanted to do this internship because I thought it'd be a good research opportunity.
Hi, I'm Lara and I'm a student at Mountain View High School in California. In the future, I hope to go into the pre-med track and study neurology when I'm in college.
Hello, I'm Christina, and I go to Hebron High School in Carrollton, Texas. I hope to major in biochemistry in the future.
Hi everyone, my name is Manya Srinivasan and I'm a freshman at Emerald High School in Dublin, California. I intend to go down the medical pathway and I've decided to join ThinkNeuro because I find medicine interesting and I wanted to learn more about it as well as getting some experience in research.
My name is Suad Abdella. I'm a sophomore at James Hubert Blake High School in Silver Spring, Maryland. I intend to major in neuroscience or biology. I decided to do this internship because I am interested in research and I wanted to get some experience.
Hey everyone, my name is Sraavani Poluri and I'm a junior attending Dulles High School. I plan on majoring in biomedical science or neuroscience, and I joined ThinkNeuro to know more and learn about how to effectively formulate research in the fascinating world of medicine and neuroscience.
So our research question is, what are the roles of inflammation and immune response in the pathogenesis of tauopathies? And how might targeting these processes lead to therapeutic intervention? Tauopathies like Alzheimer's and frontotemporal dementia feature abnormal tau protein aggression in the brain, leading to cognitive decline. Inflammation and immune responses worsen disease progression by damaging neurons and promoting tau buildup. Drugs such as GSK 2606414 targeting these processes may alleviate symptoms, while NSAIDs could potentially slow the disease. This research explores new treatments for tauopathies. In order to understand our research, we have provided some key terms that Kayla and I will go over.
First of all, tau protein is mainly found in the central nervous system of the brain. Tau protein can bind and be phosphorylated by various protein kinases. The abnormal phosphorylation of tau can lead to cell dysfunction and death.
This neurodegenerative disease is known as tauopathy. One common example of tauopathy is Alzheimer's disease.
In tauopathies, the body's immune system initiates a coordinated response to the abnormal aggression of tau protein in the brain. This immune response includes inflammation, which plays a crucial role in tissue repair and healing. One key player in this process is PERK, an enzyme responsible for phosphorylating tau proteins. Phosphorylated tau proteins are more likely to form aggregates, which can ultimately lead to cell death. GSK 2606414 is a compound designed to inhibit PERK, potentially offering a means to mitigate the detrimental effects of tau protein aggression in tauopathies.
The objective of our research is to explain the role of inflammatory immune response in the pathogenesis of tauopathies, and to describe how targeting processes like the kinase inhibitor GSK 2606414 may help create therapeutic interventions to mitigate the harmful neurological effects of tauopathies.
Using the Web of Science database, we were able to conduct literature research from the 100 most cited papers relevant to therapeutic interventions within the pathogenesis of tauopathies. We used Google Sheets and Excel to organize data and found that of those top 100 cited papers, 67 were articles and 33 were reviews. Along with Google Sheets in the BiblioShiny package, data was extracted and inputted to our programming platform to form a visual analysis.
GSK 2606414, a kinase inhibitor targeting PERK, shows potential in alleviating neuronal dysfunction and tauopathy-related symptoms with minimal side effects, suggesting its potential as a treatment option. Overactive microglia prompted by neuronal damage accumulate amyloid beta, worsening Alzheimer's progression. Furthermore, in the 34 times Alzheimer's was mentioned as a keyword, as shown in Figure 4, it was conveyed that ACT binds to amyloid beta, which exacerbates tau phosphorylation with implications towards Alzheimer's advancement.
This contributes to the progression of Alzheimer's disease pathology via the activation of kinases by inflammatory molecules. Approximately half of the 11 articles mentioning neurodegeneration as a keyword discussed how researchers have hypothesized that an increase in amyloid beta can in turn increase inflammatory proteins like IL-1 and ACT, which then increases the expression of specific kinases that lead to tau phosphorylation and neuronal degeneration, which can be seen once again in Figure 4.
A promising treatment pathway is NSAIDs, or nonsteroidal anti-inflammatory drugs, which may lower Alzheimer's disease risk by reducing inflammation in the brain. Their effects in diagnosed patients imply a role for inflammatory proteins like alpha-1 chymotrypsin, or ACT, in early stages of Alzheimer's disease, and they can also be beneficial if they're used as an early intervention strategy in tauopathies. Ibuprofen was shown to have reduced inflammatory proteins like ACT and IL-1 in transgenic mice.
This aligns with the theory that imbalances in these proteins contribute to tau hyperphosphorylation and neuronal death in Alzheimer's, and supports the connection between Alzheimer's progression and inflammation. This suggests new therapeutic approaches targeting inflammation, particularly in addressing tau hyperphosphorylation and cell death.
So this research study indicates that kinase inhibitors such as GSK 2606414 can regulate early neuronal dysfunction in tauopathies with minimal side effects. science. By targeting specific kinases, it could be a promising therapeutic strategy for mitigating the cognitive dysfunction, neuronal loss, and pathological protein accumulation associated with neurological diseases.
Inflammatory proteins like alpha-1 anti-chymotrypsin and IL-1B can induce tau phosphorylation, exacerbating the formation of tangles. It is suggested that addressing inflammation and its associated proteins may have a broader impact on the pathology of AD.
Thank you for listening to our presentation, and we hope you found the information presented as engaging as we did. Please feel free to message us if you have any other questions regarding this content. Again, thank you all for your time and interest in our research project.